welcome back to That's So Second Millennium, the Catholic Science Podcast. This is episode 94. Well, the goal has been to have an interview for the second week of the month, and a discussion between myself and Bill on a topic of my choosing for the fourth Monday of the month. Bill's a little overextended. Bill's a lot overextended. And I am in the midst of making enormous decisions for my own professional future. So for this evening, this is of course Sunday evening, I'm putting it together for Monday morning, I am going to take the 3 Maureen Condic interviews from last summer and, for your convenience, splice them all together. And I will go ahead and preface it with a little bit of the discussion here, because I have picked up and I have begun the book that she did with her brother Samuel Condic on Human Embryos, Human Persons is the title of the book. So it's their goal is to do a rigorous, neo-Thomist philosophical accounting of the deep, the intricate biological facts of what we know about embryology. I'm a little disappointed in the book so far, to be perfectly honest. We're still in the part where Samuel Condic, I can only imagine, is laying out a basis of metaphysics for us to go on with, Thomistic metaphysics, and of course so as an example, is mostly based around trying to distinguish the situation of a marble statue versus a marble block, as a sort of a continuing entity, versus a human person. And I can't possibly go into all of that tonight, because I just got back from a trip to New York. A great trip to New York, but it is, I'm exhausted, and I can't do this complete justice. I will do this complete justice, well, as complete as I can, in one of my later conversations with Bill. In any case, I am reading, so of course I'm a mineralogist. So I am reading this as an scientific expert in that a field, I know what marble is. I know a lot about what marble is. I will venture to say I know more than either Samuel or Maureen Condic about marble and its composition and its history, and I think I'm competent to have something to say about it. I don't think their example is constructed all that well. It leaves an awful lot hanging. It ignores a great deal of relevant facts. And of course it's meant to be a short introduction. And it would not be the first book that I've read that started with a short introduction that oversimplifies some things and blunders a bit, and then is followed by, when the book reaches the wheelhouse of the people in question. So in particular, Maureen Condic, being an embryologist, will know what will be able to parse out far more strictly what should be going on in the rest of the book. So I still have high hopes for the rest of the book, but that is, it is problematic. There are some problems in it so far. And that brings up, I will note, something that I have seen in my reading of the Thomists, not just Thomists, but medieval philosophers in general, certainly Thomas Aquinas being one of them, and philosophers on down to the present day in my admittedly scattershot reading of them. I think there is this compelling desire to go and philosophically analyze human beings, because of course that's what we want to, that's our goal, that's what we want to get to. I do not think the foundational work of analyzing the simpler things has been done well enough to give us the philosophical tools and instincts that we need in order to approach the far more complex and fraught issue of what is a human being. And especially to answer the modern question of how do human beings differ from purely material objects? Where is that line? Can we find that line? Do we know for certain where that line is? Are there animals that have some more rudimentary form of spirit? I mean, there's dozens of questions you could ask. This is not the place to ask them. But in any case, I will go ahead and re-provide to you the interviews that the entire interview in this episode. So this is going to be a long one. Buckle up. And if you have listened to them before and, aren't in the mood to listen to them again, then, you can skip the rest of this episode, admittedly. But it was fascinating, and she is absolutely an expert in her field, and it was a fascinating interview to talk about the biology and the moral implications from that perspective. So without further ado, once again, here is our interview with Maureen Condic at the Society of Catholic Scientists Conference at Notre Dame in 2019. We are greatly privileged to have Dr. Maureen Condic from the University of Utah here. She is the one being awarded the St. Albert Award at this year's Society of Catholic Scientists Conference. So we're actually talking the afternoon before the conference begins. So Dr. Condic has her bachelor's degree from the University of Chicago, a PhD from UC Berkeley. That must have been an oppressive environment. And she did a postdoc at the University of Minnesota. She has been at the University of Utah since 1997.
Correct.
And has been awarded a number of awards, both for her science and for her stand on life issues. So with that, I'm not necessarily one of those people who like to read a speaker's entire, we could go on for 5 minutes and read your entire list of citations. But I would encourage the listener to look them up for himself or herself. And hopefully we can just get started a little bit. So we'd like to talk today both about your personal experience as a researcher in embryology and related fields who has felt the need to stand up for what's true in difficult situations, as well as, sort of the nuts and bolts of what you do, since our audience, I think, is a number of scientists in it. And I think a number of them would be interested in hearing more about that.
Absolutely. I'm happy to be here, and I'm happy to engage all of those topics in whatever order seems to make sense. Okay.
Well, we'll do our best. Making sense is something that we work on. It's a goal that we aspire toward. We don't necessarily always get all the way there. So, yeah. So, at what point did you feel drawn toward biology and biological sciences?
It's an interesting question, because I think my vocation as a scientist happened quite early. I lived in a small town in the Midwest and was easily bored. So I remember going to my local library, public library, which is a tiny little library.
Yeah.
And after I had blasted through all of the young adult section and anything else that seemed of interest, I discovered biographies and fell in love with these little stories from history about real people, but with all the drama and heartbreak and everything else that comes along with a biography-worthy individual.
Yeah.
And being the rather OCD person I was then and still am, I started at A and I started reading through all the biographies on the shelf. And I remember vividly that, when I got to see and I read the biography of Marie Curie, I had never, ever encountered anybody who I could relate to as strongly as I related to her. And I remember just closing the book and just going, that's it. I'm going to win the Nobel Prize in Physics and Chemistry. I'm going to die from radiation poisoning. It's just my life. It's going to be like this.
It's going to be like this.
Right. Yeah. So I decided I was going to be a scientist. My father was a philosopher. My mother was a mom. And they thought I was completely insane and that if I just grew up, this had to have something to do with hormones, because I was probably about 12 at the time.
Okay.
It would wear off, but it never did.
We're still waiting.
We're still waiting. Yeah. So that's how I got interested in science. And how I got interested in biology, I think, I've often speculated that there's kind of a reciprocal relationship between the size of the question, its intrinsic interest, and the precision of the answer. So the really big questions, like, what is consciousness in the brain?
Right.
How did life evolve?
Right.
Sort of the things that you can spend your whole life pondering and that are extremely compelling because they encompass such epic dimensions. They're fabulously interesting questions, but the answers to them are inherently imprecise. You can never get a good, clean answer to that question. On the other end, if you're interested in, what the upstream regulatory elements are that drive expression of a particular gene in a particular tissue at a particular time in development, you can get down to a 10 or 11 letter answer to that that's extremely precise and very manipulable, but also intrinsically boring. Right. So this is not a compelling question, but the precision of the answer is extremely intellectually satisfying. So on that trade-off, everybody's got to find a point where the questions are big enough.
Yeah.
The answers are precise enough that you can be satisfied with those.
It's actually tractable. Yeah.
So for me, that turned out to be cells. Cells are living units. They have interesting behaviors. They're unpredictable. Mm-hmm. They have an aesthetically beautiful quality to them. You can look at them and be pleased by just their elegance and complexity. But they give you tractable answers. You can get precise enough answers that. So I had started out in psychology and trying to understand what was consciousness and how did memory work and what that whole brain thing was all about. But I eventually ended up with the development of the nervous system because that's largely a cellular process and the precision of the answers is much higher.
Yeah. You can get to the point where at the end of a 5-year period, you may actually have some answers to a set of questions you asked in the beginning.
And you have tools. You have things that can manipulate the system in the future that you can test hypotheses on a very precise level. Without ever losing, the beauty and the compelling aspect of a living unit that's responding to your manipulation. So.
Yeah, that's true. In ways that I find crystals very elegant, but admittedly they're not as complex there as cells are, with all of the things that the cell has to accomplish to get its work done. So and that was So you've kind of, looks like branched out from the nervous system to, other fields as well. How has that, so what was your PhD dissertation about?
Well, so my PhD was, as I said, sort of in neural development in a very, very simple system. And I was interested in the factors that control a very, very specific behavior of a single cell in the peripheral nervous system of this organism that establishes a very characteristic route that it follows to get to where it needs to go. So, the nervous system is ultimately ends up a big computation machine, but it develops itself without blueprint, and yet it has fabulous precision, and the function of the nervous system as a machine, as a calculator, relies on intrinsically very, very precise connections between cells. And these cells can be really, really far away from each other in early development. And so it's quite mysterious. How do we wire up the nervous system? So that was my question. How does a cell in a particular location know where it needs to extend, who it needs to talk to, and who it doesn't? Some of this gets refined by behavior later on in development, but as a first pass in the embryo, we set up pretty much the structure of the nervous system with good accuracy. And honestly, we have no clue. Even now, even after my brilliant thesis, which was, I'm sure, earth-shattering and changed the thinking in the field and all of that stuff, we're still...
Go back and calculate its age index and all of that.
We're still very, very much in the dark about how it is that the basic principles of wiring happen. So that's what I started with, was development of the nervous system, but that already put me into the general swimming pool of embryonic development. And nothing develops in a vacuum. So the nervous system is kind of connected to the development of all the other things. So
the nerves themselves have the glial cells and everything that helps them do what they do. I mean, they're a minority of the cells in the brain, is my understanding.
Yes, we have many more glial cells than we have neural cells. But because I was working in a developing system, I started getting more and more interested in embryology. How does the whole thing put itself together? And I started teaching embryology as a graduate student, and then continued on as a faculty member.
So, what was your work at the University of Minnesota? What was the focus there?
Continuation of my graduate work, but in a different system. So, I had worked originally in scientists use all sorts of, as your listeners, of course, know, model systems that have strengths only in particular areas. So my initial system was a great one for a very stereotypic pattern of neural outgrowth during development. So I was working in Schistosperma americana, which is the American locust. grasshoppers.
Yeah.
And they have beautiful embryos and fabulously beautiful neurons at the embryonic stage. But they weren't a really great system for direct relevance to vertebrate models or to genetics. So as a postdoc at Minnesota, I was transferring to a vertebrate model, so mice and chicks, mostly chicks, looking at various aspects of sensory neural development in that system.
Yeah. Okay. And then after that, you got your faculty position at Utah.
Yes.
So.
And carried on. Then moving a little bit more into spinal regeneration.
Okay.
And peripheral nerve regeneration. So that was involving more mammalian models, rat models, mice models, but continuing to work in chicks because they're a really accessible vertebrate model for the kind of cellular manipulations I was doing.
Okay. That's interesting. I did not know there was a field of biology where they were that much more handy than mice were.
Yeah, but not a great system for genetics, but a really, really good system for cellular manipulation and observation, because you can pretty much leave the embryo intact and still watch what's happening inside. Okay. So that's true of other systems as well, but very difficult in mice.
Yeah. Huh. That's interesting. Yeah, that's all these details you wouldn't know unless you actually tried to get the nuts and bolts of it to actually work. So at what point, I mean, what is your history in terms of, your religious outlook? How has that been shaped over time?
I hate to characterize myself as a bad Catholic, but I'm kind of a bad Catholic. I was raised Catholic. My father and mother were both very, very faithful people and very, very smart people. Mm-hmm. And so I started out in the faith with not a lot of questioning, because I trusted my parents. They were smart. Yeah. They believed in this, so why not?
Yeah.
And it didn't really seem to make much of a difference to me. Of course, in college, I went to the University of Chicago. I was thrown in with. And
then Berkeley.
Then Berkeley. Yeah, a lot of people who disagreed with that view.
Yeah.
And at that point, I'd already had my Maria Curie, epiphany, and so I knew that this was who I was going to be, and it. If I were to define myself, I would define myself first as a scientist. I think it's so absolutely intrinsic to the way I look at the world.
Yeah.
That everything has to be based on evidence. It has to be based on logic and reason. It can't be self-contradictory.
Yeah.
This is just how my brain works. So where does Catholicism fit in with that?
Yeah.
I think a lot of people, when they first encounter other individuals who question their faith or the way they were raised, and if they have that mindset, if they have an analytic mindset, sometimes they just cave. They cave based on authority.
Yeah.
A lot of really smart people are telling me that I'm just brainwashed and this whole religion thing is incompatible with science, and I love science and I want to be a scientist, so that means I have to kind of give up the religion thing.
Right.
Yeah, but I'm just a little bit too stubborn for that.
Right? There's at least that.
Well, stubbornness and
stubbornness has its purpose.
I think that what those kinds of naysayers charged me to do was not to bow to their authority, but to question their authority. And the more you question the authority of people who propose alternative worldviews, the more you keep coming back to, well, what is actually making the most sense of the world?
Yeah.
What vision of the world actually accounts for most of the data?
Right.
And in my experience, it's a Christian vision of the world, and in particular a Catholic vision of the world that very much endorses precisely the kind of questioning mind that promotes scientific investigation in the first place.
Yeah. And you can certainly find Catholics who don't live up to that standard, unfortunately, but they're the ones that do. And and what do you have on the other sides of the debate? That's the question.
Yeah, and I think many scientists in particular really don't question deeply. I mean, there's so I often tell this story because I think it puts things in perspective. That when I was a graduate student preparing for my preliminary exams, I'm in the field of developmental neuroscience. It's a relatively new field, and I wanted to be on top of things. So I've already mentioned the OCD thing. So in preparation for my prelims, I read every single paper that has ever been published in the entire discipline.
Yeah.
it was 386 papers.
Yeah.
I remember it made a stack of about 4.5 feet on the floor.
Yeah. You weren't reading them on PDFs at that point.
I read everything that had ever been published in the field. So today, 300 papers a month come out in the field. I mean, if I read 24/7 and did nothing else, you couldn't possibly stay on top of it.
Yeah.
And so as a consequence, inevitably people get narrower and narrower. They focus down on some very, very little slim slice because you just can't. It's overwhelming.
Yeah.
And as a consequence, many scientists feel very smart. They feel very analytic, and they truly are within their narrow
subfield.
But do they think about what was the origin of the universe?
Yeah.
If there's a causal chain for everything, what's the first cause? What set all of this in motion?
Yeah.
How do you explain consciousness truly?
Yeah.
Based on properties. What are the real differences between humans and non-human primates or other mammalian species?
Yeah.
All of these are really critical questions to the conclusion that everything can be explained by reductionist means and that there is no need for a God.
Yeah.
But does the average scientist spend a whole lot of time contemplating these questions?
Almost not.
Almost because they have no time.
There is that too. Yeah, I mean that's that side of it. But it's I mean it is a deficiency in the way we teach science, the way we train scientists. It doesn't have to be that way, but that is the way what we've drifted into. Yeah. I mean I was very fortunate to have a capstone experience in my own undergrad where we actually looked at philosophy of science. We read some Popper. We read some Kuhn. We looked at the history of geology as a discipline and its own recent history, which at that time people who were still very much active had lived through the plate tectonics revolution. Oh, wait, all these ideas we had about how mountains were built are wrong. They're actually quite wrong. And that I think that also has put some humility in the field in a certain sense, like, which is wearing off now, as it inevitably has to. But not everyone gets that by any stretch.
They don't, and I think particularly in biology, there's such an intoxication with success.
Yeah.
We've had so much phenomenal success in understanding the basic underlying mechanisms of life and applying it to human disease and to drug design and to all of the things that have been so amazingly powerful in the last 20, 30 years of science. Within the field of biology, people are very confident that they're on top of their game, that they can answer all the other questions and all the questions that this weak and small-minded people ponder as they cling to their guns and their Bibles.
Right.
We don't really have to think about it.
We don't have to worry about that
because we're changing the world here and making progress and redefining the species and curing disease and. What more do we need to do?
Right. Yeah. For the people that we choose to let into the sphere of people whose problems we care to solve, as opposed to the people we're willing to use in order to solve the actual problems. But, So we talked about it a little bit before we started recording. What has been your experience as being, someone with this background and then making your way through the academy and the larger political world that's connected to it? I mean, obviously, you're here to be awarded partly because people believe that you took a very courageous stand against people and have taken a lot of bullets.
Yeah, I suppose I have. I tend to not think of it in those terms. I think at every step where I've exposed myself to the ridicule of my profession and the inevitable consequences of that ridicule. Yeah. It's always been for a reason, and the reason always had to do with upholding the truth. I mean, the main event that got me started in public advocacy and public education, which is definitely the area in which I've taken the most. Where I've earned the name pariah in the eyes of my colleagues.
Right.
All started because back in 2000, I had some really strong results on the topic of neural regeneration, of adult regeneration. And I'm not a physician, I'm a PhD scientist.
Yeah.
But the NIH picked up this result and it ended up getting into the popular press. And suddenly, from all over the world, I started getting phone calls and emails from spinal injured individuals who wanted to know if my results were going to get them out of the wheelchair.
Yeah.
I have, at that time, did not appreciate, but I have come to appreciate, and I still work with this community of patients, how very, very aggressive they are and very well educated, and really self-advocates, go out and try to find answers to their problems.
Yeah.
So I developed kind of a standard reply. This is basic research. I'm very optimistic that it may someday translate into, and that satisfied most people. Until I truly had an event that changed my life and put me on a very different course of action. And you'll have to apologize if I cry about this because I often do. I got a phone call from a young man whose wife and 5 children had been hit by a drunk driver. And his wife and the 3 older kids died at the scene of the accident, and the 2 little babies who were both under the age of 5 were high-level cerebral, ventilator-dependent quadriplegic.
Yeah.
And he's crying on the phone.
Yeah.
He's just begging me, I'll do anything. I'll give you my own body. You've got to do something for my kids. And I'm crying. I had no idea what to say to this man. And at some point, I asked him some neutral question about getting support or, trying, who has he talked with? And he goes into a complete diatribe against George Bush because his senator had contacted him and was trying to convince him to bring the girls to Washington to testify before Congress about stem cell research. And his senator had told him that there was a cure that was being blocked by George Bush's policies on stem cell research.
Okay.
And the rage, just blind, frustrated rage of this man.
Yeah.
I remember it hit me like a bolt of lightning. I'm looking literally at the phone.
Yeah.
And going, nobody who suffered as much as this person should be used like this.
Yeah.
Should be used as a political pawn.
Yeah.
Because that's all it is.
Yeah.
The senator knows that if he brings these poor wheelchair-bound babies to Washington, that they're going to get political support for a research agenda that he's simply willing to lie about. Yeah. And so I decided right then and there, somebody has to tell these people the truth. Has to be willing to spend time and effort and develop a rapport and talk with people about what we really do know. And so we talked for 2 hours. And I think it was the hardest conversation I've ever had.
I'm sure.
And in the end, he thanked me. It was a very sobering realization. I had to tell him that there is no cure. Nobody is hiding this from you. This is an incredibly complex area of research. It's moving very, very slowly. Stem cells could potentially have some benefit here, but in actuality, it wouldn't be in my top pick of the top 500 things that could be helped by stem cells. Right. Your girls are going to be in a wheelchair for a long time. And your job is to try to help them have the full life they can have. Yeah. Because there is no secret cure. Yeah. And, I set out from that point forward trying to get the word out and trying to be an advocate for patients and for knowledge and for factual information. I think some of the problem with science is we start believing our own press releases. Right. And people really have, the scientific community has such optimism for the power to cure, the power to move forward, that if we just unfetter science and put the least number of restrictions on people, we're going to have the highest benefit. But there's a difference between saying that and having the data to support it. So that's what I've tried to do, and I do it a lot. I fly all over the world and talk to people about what we know, what we don't know, what we're unlikely to ever know, and why.
That's a tall order.
Oh well, we all do our part in some way, but I think the more you do it, the easier it gets. You find ways of speaking to people that bring with them and that help them wrap their heads around these very, very complex topics in a manner where they can assume the responsibilities of responsible citizenship. They can influence the political process in an appropriate way without relying on scientists who have conflicts of interest and a vested interest in having things come out in a particular way.
Yeah. That's one thing. I mean, once you get how people actually live their lives involved in it, then the stakes go up so much, and it becomes so much harder to do science, which demands, that willingness to say, you know what, I might be wrong about this.
That's very true. Absolutely true. Especially when you've invested so much of your life and so much public money and the effort of your trainees and everyone else, and being willing to accept no for an answer is a tough job.
Yeah. If I just get a few more data points and I try another couple of statistical analyses, maybe I'll get something that makes it look like it's a success. It's a temptation. That's a difficult temptation to turn down.
Where does that whole world of stem cell research stand now? Are you finding yourself saying this is possible more often now, or are expectations even rising so that you have to still say no a lot of times?
I think one of the things that made stem cells kind of a low-hanging fruit for criticism of the outrageous scientific optimism that we were going to cure everything from baldness to ingrown was the fact that there were such obvious, long-standing scientific problems associated with the application of stem cell research to patients. And the whole nature of serious, long-standing scientific problems is that they're serious.
Right.
And long-standing.
And someone would have fixed them if they were that easy to fix.
If they were easy to fix, they would have been fixed. And very often it's because the problems are not just complex, but they are linked to the intrinsic nature of the cells themselves. So one of the problems I pointed out in my first critique of stem cells being the panacea for all human disease was the reason stem cells are scientifically interesting is that they have the capability of developing in a wide range of different developmental pathways. Right? So if I take a skin cell from you, it's pretty much restricted to being a skin cell, and it's hard that we have found ways now to make it do something else. Stem cells are great because they can produce all the different cells within the body. And yet, this very capability that makes them interesting scientifically also makes them extremely problematic medically, because one of the ways that evolution has protected the DNA of a cell from accumulating random mutations, which would turn it into a cancer cell, is to take all the DNA you're not actively accessing. So if you're a liver cell, you really don't need all those genes that control heart function or skin function or brain function. And so this is a simplification, but a useful way of thinking about it. So you kind of pack all of that DNA up in a chromatin structure that is much more resistant to mechanical and chemical insult by essentially inactivating it. So that's what makes cells stable enough to persist without accumulating mutations that will turn them into cancer cells.
Right.
But if we don't do that, if we are the kind of pluripotent stem cell that everyone is very fascinated with as a biological question, that cell is intrinsically prone to accumulating mutations and becoming a cancer cell. This has been shown hundreds and hundreds of times that inevitably these cells always undergo some kind of chemical mutagenesis, structural mutagenesis. They turn into cancer cells very, very readily and very reliably. So you might remember Richard Nixon. You're old enough.
Thank you for wondering.
Okay, and you remember that, back in 1971, he started the War on Cancer at his National Cancer Institute, okay? And since then, we have spent hundreds of billions of dollars. Countless crews of brilliant scientists and physicians have been devoted to addressing this problem of cancer, oncogenesis. How do cells turn into cancer cells? And it's not like we've made no progress, but if you look at the death rates from cancer over the last 50 years. Smoking deaths have declined dramatically, or because people cease smoking.
Right.
But cancer deaths have, we barely need a notch.
Wow.
And it's because it's a tough problem to fix. So, honestly, given that we've made such limited progress on these questions since I initially made my predictions that we weren't going to progress rapidly towards the application of stem cells to medical conditions, nothing has changed. I should change my middle name to Nostradamus, but but all the predictions I made back in 2001, 2002 have really been upheld precisely because they weren't just frivolous naysayer objections.
No,
they were things like, these are foreign cells. They're going to be rejected by the immune system. We've known about immune rejection for many, many decades. Yeah. We've made some progress, but it's still an enormous problem, and people still die from incompatible tissue transplant.
Yeah.
This oncogenic transformation thing, which is very, very well established. The other thing that's challenging about stem cells is even when they're not cancer, they are tumor forming. In fact, that's the gold standard for stem cell biology. If you develop a cell line and you imagine it to be a stem cell line, a pluripotent stem cell line, the gold standard test is you inject it into an immune-compromised mouse and you wait for the mouse to die because it has tumors all over its body that are formed from the stem cells, and the tumors contain derivatives of all the major embryonic lines, and that's how you know you've got a stem cell.
Yep, we were successful.
Yeah, we were successful. So let's put those into humans. Yeah, let's do that.
Brilliant. Yeah.
So these are so oncogenicity, the tumorigenicity, the immune incompatibility, and the final one is you could fix most of these if we could control the development of these cells into more mature derivatives. So if you are a patient who has a heart problem, we want to create heart cells for you. If I could start with stem cells and drive them up to mature heart cells that we could use for transplant or treatment, then they would no longer have these problems. They wouldn't make tumors. They wouldn't be prone to
teratogens, or whatever you call it.
Right, and if we took the cells from you, or if we had a big enough bank of cells, we could perhaps address the immune problem. But we've been working on stem cells. The first mouse embryonic stem cells were isolated in more than 35 years ago, and driving them into mature states is very, very challenging. I mean, it's not a simple problem that we're just going to fix. Throw a few $1 million at it, and it'll be something we'll figure out. So all those problems,
10, 20 doctoral students will get this knocked out. Yeah, we'll knock this right out.
So it's not that it's not an interesting area of research. I think it's a fabulous area of research. I work on stem cells.
Right.
I am not opposed to stem cell biology, but transitioning basic research into medical applications faces many significant problems, and those problems still stand.
And is there still that polemic between embryonic stem cells and as a layperson, I'm not sure, is that the pluripotent stem cells?
The embryonic stem cells versus what people generally just call adult stem cells, more tissue-specific stem cells.
Exactly.
So that dynamic has really, really declined because over the last 10, 15 years, the clinical applications of stem cells derived from mature tissues, so-called adult stem cells, have proven to be really, really strong. I mean, they're treatments; they're not cures. We don't have magic here, but we do have thousands of clinical trials involving
adult stem cells.
And that are showing benefit to patients. And so, I think the other big new player in the field, of course, is the reprogrammed cells that Yamanaka won the Nobel Prize for. Those cells have proven to be wildly popular among scientists because they're easy to make. They sidestep all of the ethical issues. We don't have to worry about getting eggs. We don't have to worry about getting embryos and then destroying those embryos. But they're a lot simpler. Honestly, we don't have to have a relationship with a fertility clinic who's going to give us spares and right. So definitely that has been a game changer in the field as well.
So, what was that the specific name of that last type of stem cell?
The induced pluripotent stem cells.
Induced pluripotent,
cells. Yeah. Amazing. One of the most important scientific findings in the last 50 years, I think.
Yeah. And how long has that? It seems to me like that's been a few years since that was first.
2007 was the first demonstration in animals, and then 2008, I think, or 6 or 7, I can't remember. Animals, and then human tissue, and human neural parts in 2012.
Yeah. I thought it had been a minute. So that's a beautiful thing. So I'm staring at Bill, and Bill is also this sort of bad influence on me. At some point during your discussion of the problems in dealing with embryonic stem cells, I'm going to date myself, not quite back to Richard Nixon, but I've spent way too much time watching the 80s Star Trek movies. And so there is this parallel between, if you remember, the plot of Star Trek 2 and 3 revolves around this Genesis effect. Yeah. Yes, the Genesis planet. So Kirk's old girlfriend is creating this thing that will, terraform, this planet and create life. And then they realize that the planet, in Star Trek 3, they realize the planet they've made is unstable. Mm-hmm. And they're asking Kirk's, son. It says, illegitimate son, as if we care about these things in the enlightened 23rd century, I'm sure. And he's like, well, we were using proto-matter. And somebody, not Spock, because Spock is dead. We're bringing Spock back. I think McCoy or somebody says, that's totally, that's against Federation regulations. It's too unstable to work with. And he's like, yeah.
We wanted to see if we could do it.
Yeah. We wanted to see if we could do it. Yeah. And it's it happens to create planets that self-destruct and, consume Christopher Lloyd when they go, which is a tragedy. Terrible tragedy. But, we've talked about these terms, and I was doing a little bit of reading, not as much as I would like to, of some of your work before putting this together. So we've used the word, I believe, pluripotent already, and you have used the words totipotent and plenipotent in print as well. What if we could break down what the definition of just those three terms are? They're sort of
I did coin a new term, which I did with great trepidation, but I did it precisely to try to get around an incredible misunderstanding in the public and a misuse of terminology by the scientific literature. Yeah. So totipotent is a term that means having all powers, able to do everything. Yeah. And the scientific literature uses that term in two distinct ways. Mm-hmm. The first one is the ability to make literally all of the cell types that come from that one cell embryo. So this would include all of the cells of the embryonic organs of the placenta and embryonic membranes, as well as all of the tissues and cell types that exist in your mature body. So if you can do that, you are totipotent. You are the kind of cell that has the ability to make everything.
So the zygote is totipotent.
The zygote is
the gold standard.
The zygote is totipotent, but so are many stem cells in this limited sense, in the ability to make all of the cell types of the body. Mm-hmm. So in mouse, for example, stem cells taken from embryos are pretty much restricted to making just the cells of the mature body. So they don't make the cells of the placenta, for example. Right. But human embryonic stem cells actually have the capability of making everything. So they're totipotent in that simple sense, the ability to produce all of the cell types that are normal human cell types. Mm-hmm. The stricter sense of totipotent is the term as it would apply to a one-celled embryo. So a cell that has not only the ability to make all of the cell types of the body, but
in fact,
organize
it's going
them.
In
a coherent, rational sequence that allows for, I can't say rational, but in a coherent sequence that allows for the ordered production of increasingly mature states of human development. Right. So if you have that kind of totipotency, so if I take stem cells, for example, and I aggregate them all together, they can and do produce all the cells of the body in a chaotic mass like a tumor. Yeah. But an embryo will produce all of the cells of the body in an ordered developmental sequence that progressively moves towards more mature states of the human form.
Yeah.
So that kind of totipotency is the kind of totipotency that really is all powerful, can do everything. Pluripotent, in contrast, refers to cells like I just described, the mouse embryonic stem cells. So cells that have the capability of producing all of the cell types found in the mature body, but not all of the cell types found in human development. So the cells of the placenta and embryonic structures that are transient. So pluripotent is able to make all the cells of the body. Totipotent, in the strict sense, is to be an embryo, is to make all the cells of the body and also organize them in a coherent body plan. And we needed something to replace that simpler sense of totipotent, which is confusing when people use it, because people will read that and say, oh, they've made embryos. And what they've really done is made a tumor that has this broader developmental capability of producing all of the cells that are possible to be produced. So that would be pluripotent.
That's fascinating. That is a whole world of, when I early on decided to go the opposite direction of biology, it's like, oh yeah, I'm leaving that behind, aren't I? So that does bring up the question, which I believe you've addressed at length in different places. At what point because of course there's the phenomenon of twinning, of identical twinning. At what point does the zygote, at how many divisions down does a human, as opposed to a mouse or a turtle or a chick or whatever, how many divisions down does it lose the ability to be fully totipotent in that sense?
Yeah, this is a really, really important question. In fact, I have a book in press at the University of Notre Dame Press called Untangling Twinning.
There you go.
That addresses both the philosophical and the biological issues associated with twinning in great detail. So the short answer to your question is proving a cell is totipotent. It takes a very strict scientific test. You need to isolate that cell from all other contributing sources of information, and then you assess what its capabilities are independent of any other cell type within the embryo. Now, the hard thing about that experiment is that cells don't like to be isolated, and it's very hard to do it in a manner that you don't get the negative finding of the cell dies, or it doesn't do anything, it just sits there. So when that happens, you don't really know what its potency is, because the only way you can assess it is if you get a positive finding. Right. So with that as a caveat to looking at the strict question of totipotency, the ability to not only produce but to organize all of the cell types in the body, there appear to be species-specific differences in how long that capability is preserved. So in mice, I think the weight of the data is that the first two cells produced from the zygote, if separated, can go on to produce independent little twin mice. But after that, you lose the ability to do that. In some species, for example, cattle, for some reason, seem to preserve totipotency up to, say, the 4-cell stage. Pretty routinely. So people clone cattle fairly frequently by blastiner separation, by separation of the early first 4 cells of the embryo. And each of them can routinely go on to make a little baby cow.
It's progressed to the calf stage.
Right. There's one unreplicated report in a particular species of pigs that asserted, that I would just call this an assertion because it's never been shown by anyone else, that in that species you can preserve totipotency up to the 8-cell stage. So each of the first 8 cells, if separated, could potentially produce an embryo. But I think in the vast majority of mammals, the 2-cell stage is about as far as you can go. I think cattle are really the exception. I think this one report in pigs is probably not correct.
Happens sometimes.
It does happen. Or it was an almost finding that nobody's been able to replicate since then. But that's not the only way that twinning can happen.
Then there's fraternal twinning.
There's fraternal twinning where you have 2 independent embryos and they just so happen to coexist in the uterus at the same time.
They have to be womb mates.
But there's also the capacity to develop as an entire embryo, as a whole, unified whole, can exist in a multicellular embryo as a whole. No one cell, if you took it out, could restart the whole process of development all over again.
Right.
But the entity as a whole has that capacity. So, and there's very good experimental evidence and some evidence from fertility clinic work that splitting an embryo much, much later, an embryo that contains hundreds of cells, will cause each of the halves to heal and go on in development to make twins, identical twins.
Yeah.
So that kind of splitting, that isn't separation of the earliest cells, but an actual division of the embryo in half. That can occur up to about 14 days of human development.
Okay. So we have some evidence that happens naturally as well as, or at least we have no reason to believe it doesn't happen both at the two-cell stage as well as.
We have reason to believe that it never happens at the two-cell stage. Although experimentally we can do this.
Okay. But all human, in the wild, identical twins.
Identical twins. I'm going to argue, I argue in the book that I think the vast majority, if not all, identical twinning in humans in the wild, so to speak, in natural situations occurs at the blastocyst stage, which is a stage where you have many hundreds of cells in the embryo. Yeah. It looks kind of like a hollow ball.
Yeah, because it has both the wall of the placenta that's forming as well as
the inner cells that are going to go on to produce the postnatal body.
Yeah.
So all of that early development happens inside a protein coat known as the zona pellucida. It's a very rubbery, tough coat that protects the embryo and the egg when ovulated, and then the early embryo as it's traveling down through the fallopian tubes to the uterus. But in order for it to implant, you have to escape from this rubbery coat because the outer cells.
And that rubbery coat is also generated by the embryo.
The rubbery coat is actually generated by the egg as it's developing. So it's a structure surrounding the egg. Oh,
is that different from the corpus luteum?
Yes.
Okay. Yeah, there's so many things.
All this terminology. Yes. The corpus luteum is a structure within the ovary. Part of the organization surrounding the maturing egg called a follicle.
Right.
And so the cells of the follicle that remain in the ovary after ovulation are the corpus luteum.
Yeah.
But the zona pellucida is this proteinaceous coat. It's very simple. It consists of 3 proteins, creatively called zona pellucida 1, zona pellucida 2, and zona pellucida 3. But they make a nice, tough, protective coat for the egg. The sperm actually have to penetrate through the zona in order to get to the surface of the egg to have fertilization happen. And then all of early development happens within the zona. But in order for the embryo to implant, the outer cells actually of the embryo actually mediate implantation. And they have to escape from this covering, or they can't interact with the uterus.
Yeah.
So this happens through a process called hatching. And I have some great movies of this. It's just unbelievable. It really does look like the embryo secretes some enzymes that weaken the zona A1 region. It's usually a small tear, and then this entire structure will squeeze out of that little eggshell.
Yeah.
And people have observed twinning happening at that exact point.
That would be a point where you would expect that to happen.
Two little bubbles instead of one little bubble.
Yeah.
And so long as each of them inherits some of the cells in the inner cell mass, you get identical twins.
But are there times when it divides in a less fortunate manner, and then you simply unfortunately visit
one that just has some cells missing, but they're not sufficiently large enough group of cells for them to carry on the development on their own and regenerate.
Oh, that gets complicated really fast.
But I think part of the confusion about twinning is the belief that twinning happens at the 2-cell stage, and we can do this in the laboratory. People do it routinely, but it has never actually been observed.
Okay.
Despite the fact that in IVF situations we have daily observations of embryos, many hundreds of thousands of embryos have been watched over many years, and no one has ever reported this happening.
Nobody has seen that happen. Okay.
Even though the actual rate of twinning in IVF situations is highly elevated compared to natural.
It's what? It's higher.
It's quite a bit higher.
Quite a bit higher. Yeah. So in that situation, which no one should be exposed to, but that's the world we live in. So that whole, and I forget the name of it already, but that protein coat is present as well, and that's growing in the dish or what have you. And So that has to swell, and then at some point that gets implanted, and this whole tearing process could also happen. I mean, there would be a more or less normal rate of that style of twinning. Well, I mean, that would be, no, you're saying it's higher.
Yeah.
And that's when it would happen.
Yeah, and some of the thinking about this from animal models is that the longer period of time you have the embryos in culture, the tougher that protein coat gets.
Yeah.
And so the likelihood that you're going to have a really constricted aperture through which the whole embryo has to escape would potentially increase the likelihood of twinning.
Yeah. The adventures that we all have to go through without, before we have any chance to remember any of it.
Yeah, it's fascinating. I've often been asked how, my experience as a scientist was, has given me a better appreciation for humans and for human life. And, honestly, it wasn't until I was pregnant and I really started thinking about what I knew about embryology in a practical sense and in a personal sense that I became completely flabbergasted that anybody ever survives to maturity.
Right? Right?
Just how can this ever work?
How does this? It's all got to go just right.
Exactly, and there's so many variables, and they're all having to happen exactly at the same time, in exactly the same way. And oh my gosh, it really is. It does give you a sense of awe.
Yeah.
And
without question.
The miraculous nature of human development that we ever get out here more or less intact with 5 fingers and 5 toes.
Right. Yeah.
Given that whole realm of awe and wonderment and amazing unlimited potential for different things to happen at that stage of life, I have to just think as a Catholic layperson that, in the Catholic imagination, it seems so viscerally sad and disturbing that this wonderland, in a sense, is so easily dismissed as accomplished cells.
Yeah. No, there's that. I was thinking the side of we invade it and manipulate it to our own ends so readily, but that side of it too. Yeah.
Both of them are disturbing, and also I find that attitude among my colleagues. I don't want to say disingenuous because I honestly think they don't think about it, but it is a distortion of reality and of a reality that they should know better.
Yeah, and you spoke early on about that. What compelled you early on was the big picture question, and what you've been discussing here is a lot of big picture, big possibility kinds of questions. And presumably you're not alone among these biologists. They too were drawn in by big questions, big picture at some point in their early lives. And yet it falls by the wayside, and that too is viscerally sad, it seems to be.
I don't know if anyone's ever done a sort of sociology study of what draws people to science, because I'm not sure I would endorse the concept that most scientists are drawn to it by big picture questions. Really? At least not in biology. Well, I think in biology, the vast majority of people who are drawn to it are people who, took their mother's toaster apart when they were 8 years old and confident that they could put it back together, except for that last spring
or something.
It didn't quite fit.
Or the thing that they crossed it and shorted it and sort of got the Pop-Tart on fire.
You might have had a toaster experience in your past.
It wasn't a toaster per se, but some things of that nature.
Okay, well, so I think biologists in particular are not people who spend a lot of time contemplating the big pictures. They're not like cosmologists or physicists or maybe geologists who have this huge.
There are different branches. Yeah, I mean, the people who go into oil and gas are, they have an end and a means to that end, as opposed to the people who study the history of the planet as a whole. Yeah. Or planets now, multiple planets as a whole. Those are fairly big picture questions.
Yes, I think that in my experience with biology across many different organisms and different orientations on more or less pertaining to medicine or to basic science, the really unifying thing is the tinkering mentality. I want to be able to take things apart. I want to put them back together in cool ways. I just want to see if I can do it. If I can take that toaster apart and put it back together. And I'll just, again, I love to tell stories. This is how I order my mind, I think.
We're doing a podcast, so this is exactly when you should exercise that. That's excellent.
So I had a postdoctoral advisor who was a fabulous fly geneticist at Berkeley. Really just the kind of mind that you could just stand in awe of. I mean, he would lecture, and pose some biological question. Mm-hmm. Have you ever wondered how gene X interacts with gene Y? Mm-hmm. How would you test that? Right. And then he would just, off the top of his head, describe these incredibly complicated genetic processes. Everybody's frantically taking notes, and I'd go home and an hour later work out. I guess that actually would do what he said it would do. But he was so experienced.
The logic of
so logical, he could just do these things off the top of his head. But and amazingly, as many scientists are, a confident individual. And I remember more than once, after going through one of these very, very complicated genetic processes to test a particular idea, he would pause dramatically with his hand on his hip and his other hand on his chin and say quite smugly, "So, I made that fly." I made that fly. And the fascinating thing is that attitude. No, I didn't test the hypothesis. No. I didn't discover the information. No, I had an idea in my head, and I manifested it in reality. Yeah. I made the fly. Right. And that attitude, it's tinkering writ large. It's the ability to have an idea and to make it happen. Not to discover what's in nature, but to make the fly yourself.
Yes, it's not discovery. It's a creatorship. It's would it be hyperbolic to call it a kind of God complex?
Because it sure sounds like that. Yeah, I mean, that's the whole...
Well, but that kind of power, that it's so intoxicating. I mean, people who love the experimental side of science, who, biologists I know, myself included, would wake up in the middle of the night and look at the clock and go, okay, the experiment's been running for 4.5 hours. If I ran into the lab right now, it would be gone enough so I could tell if it worked or not. And they would get on their bicycles, drive to the lab at 3 a.m. because when it works, it's such an incredibly powerful feeling of, you had an idea and you made that fly. You made it happen. In reality, it isn't. And it almost is completely stripped of any sense of discovery. There's almost no wonderment about it. It was that you were clever enough to make it happen.
It's sad to lose that sense of wonder.
Yeah. Really. Well, it's sad and sort of contrary to the scientific enterprise. Yeah. We consult reality and see what it has to say. Well, I mean, and of course, there's the whole, there's both a philosophic and a spiritual side of, you were able to make this fly because there were already flies. Right.
Yes.
Everything from the fact that there are things rather than nothing, all the way down through the existence of laws of physics that allows complex phenomena like this to happen, to the construction of a genome where there's already all of these pieces in place, and you took some bricks out and slid them in different places and something cool happened. And that's great. But
it is, and one of the reasons I tell the story is because of the obvious absurdity of that claim.
Yeah.
I would often push back on him. Oh, so what chemicals did you start with? When you made the fly, I would love to do that myself sometime.
Yeah, exactly. Yeah.
I'd make a really big one, so I could fly to work on it.
There you go. Exactly. Yeah, that's right. I'm gonna go down. I'm gonna buy some coal, and I'm gonna purify it, and I'm gonna get the carbon and hydrogen out of it, and I'm gonna make a fly. Let's start from there.
Right. And it's odd because again, they're so poor. There's such a complete lack of any kind of philosophical training for the vast majority of scientists. They just don't taught these things.
Yeah.
But it struck me in your litany of everything from the Big Bang all the way up through to the existence of a genome.
Yeah.
I would say right before that, you still have to have the existence of the soul. You have to have a substantial form for any living thing that is kind of the gravitational forces towards which all of those biological processes are working, and that hold them together as a coherent thing. So that when you take out a few genes, sometimes you kill the fly, but you rarely cause it to break down into carbon. That result was never going to change.
It hasn't gone all the way back that far. Yeah, that is. That's fascinating to me, and I don't know enough Aristotelian Thomistic philosophy to do this yet. Maybe I'll be crazy enough to, go off in that direction. But old Aristotle and his form and matter idea.
Yeah.
It seems to me there are a lot of ways it works better with modern science than it did when you were trying to say, well, it's a table. What's the form of a table? Well, it's kind of has to be sort of like this. Whereas, what's the form of an electron?
Right.
Well, that's pretty precise.
Yeah.
What's the form of Drosophila melanogaster? The DNA, the genome is a fairly precise set of rules. It's not that hard, but then that would be only part of the form.
Well, it's a very so prior to this book that's coming out on Twinning, I wrote a book with my brother Samuel, who is a philosopher, and to my knowledge, this is one of the few books that has really attempted to bring to bear onto the question of anything, in our case, the embryo, a rigorous philosophical analysis and a rigorous biological analysis. And the interesting backstory to this book was that the hardest part of writing it were the 3 or 4 years where we just had to fight endlessly over terminology. Like, what do you even mean when you say this?
Right? Yeah. And that's one of the most generous things you can do is to put the result of that argument out there for other people not to have to have the whole argument.
We were both convicted that we were talking about the same world. And if you're talking about the same world, you have to develop a common vocabulary that allows you to talk about it intelligently and a common shared set of understandings.
Yeah.
But as a result, when we had finally worked through mostly an education on my part, learning biology is relatively simple, but wrapping your head around hylomorphism
said the biologist.
Is much more challenging. Yeah. I think that the amount of information you have to acquire to wrap your head around biology is enormous, but the complexity of the ideas they're all fairly trivial.
Yeah.
Overall,
a very large number of relatively small inferences to get to where you're going.
To get to whereas trying to understand what is the form of a Drosophila is a much harder thing to do. But I would say the second biggest fight was once we had gotten to the point where I could barely hold on to all of this and think about it intelligently, I'd insisted, well, we have to have a chapter in the book that addresses this from the perspective of for people like me, people who never really thought about it this way before. Yeah. And to my brother, that was just beyond. Everybody knows this. Of course, it's obvious. Why do we need to talk about what hylomorphism is?
The dangers of specialization. Yes.
So the first chapter in that book is, I think, a great. It reflects me, the ignorant scientist, trying to. This is my understanding. Endorsed by somebody who has a much more natural sense of the relationships between the various components, and it writing that chapter really convinced me that intrinsically all scientists actually are Aristotelian. They look at it that way. They're Thomists. They have this understanding. We just don't know they have it because you can't really do science without an understanding of the fact that the subject of your experiment can change and remain the same thing that it was, despite change. Yeah. Because if we didn't have that, we would never be able to do an experiment. We'd never even be able to interpret our results because every incidence would be its own independent thing.
Right. Yeah. What is the title of that book?
That book is called Human Embryo, Human Being, a Scientific and Philosophic Analysis.
Right. Is that important that very few books like that have been written that attempt to synthesize the philosophical and biological debate?
I think a lot of people try to do it, but they do it very poorly because they don't go through that 3-year process of really working to try to understand or work with another person of sufficient competency. So you have a lot of philosophers who dabble in biology and come up with some grand unifying theory that is completely
doesn't really contact the truth on the ground.
Well, it's completely uncoupled from the facts. Except for sort of the high school level facts that maybe are 40 years out of date and overly simplified.
Yeah.
And then you have a lot of scientists who, get old, get tired of writing grants, and suddenly imagine themselves to be philosophers. Hey, I wrote Contours back in high school. And,
right.
I picked up a few modern,
I think Bertrand Russell or someone.
Yeah, that's right. So I'm confident, and now I'm going to speculate away about, the meaning of the universe because, I'm old and crotchety, and I know I'm smarter than most everybody else I know, and so I should be allowed to do that. But of course, they do it very, very naively and completely uncoupled from any kind of philosophical rigor or because it's hard.
Yeah,
it's hard. I certainly couldn't have done this on my own. I mean, we had a true collaboration, and it was I think very certainly for me a very productive one, and I hope a useful one for everybody else.
Yeah, interesting. I mean, and to take that and I mean, that is the thing is. Yeah, there is like you said, there is so much philosophy out there that sort of attempts to, and it's sort of glibly, of course, from my own specialty, I can look at things, are you really defining that? From the perspective of geology and chemistry, that's not necessarily an adequate treatment of that. And that's, and then of course, the other side of, the scientists who even bother to convince themselves they need to write something about it. Gotten on that opinion of themselves. And that seems to me completely, I mean, that would be what's required to build that bridge to, it was what we would have to do now to capitalize on the philosophy and the science that we have, the way that it was, and you can't replicate it, and it would be much more complicated. Thousands of people instead of the several dozen that probably gave us the medieval synthesis in the 13th century. But what work could be, I mean, to me, even if you are, even if you're just laboring away trying to, okay, how am I going to apply this to the question of what is a tectonic plate? or crystals or minerals, what should we define as a mineral species? Where should we draw those lines? Should we have all these names? Even just that. But that would still be, I mean, so I use this example, from earth science. If you've seen 3 tin mines in Cornwall, you think you know how tin deposits work. And then you might go to Spain and you're like, as it turns out, cassiterite can crystallize in this completely different environment. That's interesting. I better do some more work and see if there's a common. And then go to Indonesia. And by the time you've got 30 tin mines, you might not be as surprised when you go and see tin mine number 31 and say, okay, this actually is at least like existing patterns. And that I don't know that there's a need to go deep and there's a need to go broad. There's also kind of a need to be at that 45 degree angle in between. And I think that's really where there's stuff lacking in terms of, I think the philosophical ideas would get a lot more rigorous if we exposed them to enough different environments and demanded an actual, what would work with the facts on the ground and this science and this science and this science. And you would start to see patterns of things that I think we're missing.
I agree.
Some idea.
And I think we have this throughout the scientific enterprise. As someone mentioned earlier, the need to, the demands of science to falsify your own hypothesis.
Yeah.
Is a really, really tough thing to wrap your head around. And in practical fact, people tend to shy away from looking at evidence that could disqualify their hypothesis.
Yeah.
And that even applies to things that they've never really formulated that good hypothesis on. Everybody knows.
Right.
And so let's not actually look into the literature and find out if that's true or not, because I'm comfortable with everybody knows. Because it supports, whatever vision of the world I want to have and allows me to just assume.
Yeah. And does that phenomenon in the scientific world have an analog in the political and policy world, such that people don't want to...
The phenomenon in the scientific world is a subset of the general human tendency, the legacy of original sin, whatever you want to call it.
Right. But it does seem to help explain the intractability of things like the abortion debate.
I think, not that we should open up the topic of abortion, but I think many things contribute to this, including people's unwillingness to look squarely at the facts, people's willingness to immediately turn a presentation of the facts into an ad hominem attack.
Yeah.
That if you're going to assert that a human embryo is a human person or a human being, you're just saying that because,
right?
Because you're Catholic, because you're an ideologue, because
you're a hater or a shamer.
That's right.
Yeah.
So we can throw a name at you because you said something that makes it challenging for us to maintain the position that we'd like. But that's not all of it. It's not like people wake up in the morning and suddenly find themselves a pro-abortion person. I mean, people wake up in the morning and they find themselves a person who's very, very sympathetic to women and wants women's freedom to be optimized and wants to make a level playing field in the economic world.
Yeah.
Maybe they wake up and find that somebody they know is in a crisis pregnancy and doesn't know what to do and is suffering. That's how people wake up.
Yes.
And they formulate their views on abortion based on those experiences, not on the facts.
Yeah.
I mean, and those are different. Well, I mean, not all of those are factual, but the situation of waking up and yes, this person I know is in a crisis pregnancy. Yes. And what are all the societal factors that went into governing why she had a sexual encounter with this person when she doesn't want to be pregnant? What causes that? And we don't want to look at that either.
Well, it's just tough. I think the ongoing debates over complicated topics like this are, I'm fond of saying you can't reason people out of a position they didn't reason themselves into in the first place.
No.
And when people find themselves supporting a particular position out of emotional life experiences and out of conviction. And out of beliefs that perhaps they've never really questioned, you can't laying out the facts for someone like this doesn't help them.
Yeah.
But is there still real value in having those facts?
Oh yeah,
laid out.
Oh yeah, I do my best. I fly all over the world, and for food. trying to talk to people about these topics because
we'll talk for food and plane tickets.
Food and plane tickets, yes. Because it does help people to have the information laid out for them. And on this particular topic, on the question of when life begins, there's actually a relatively simple and uncontroversial answer to it. A lot of the controversy gets manufactured when you move up from the question of when does human life begin to the question of when does that life have value? Mm-hmm. When is it subject of rights? Yeah. And that's a political question, not a scientific question. And it's an ethical and moral question. And that's really where the rubber meets the road. I mean, I think that it's actually been fairly encouraging in the last 15 years that the scientific arguments about this used to be very, very shrill. A lot of people asserting there's no consensus in the field and no one knows, and fertilization is a process and development is a continuum, and yet a lot of that has dropped away. And even the most honest people on both sides are willing to say, yeah, sure, it's a human being. But we deny rights to human beings all the time.
That's right.
We do. And so maybe this is a case where the rights of the mom trump the rights of the fetus or the embryo. And that to me is a step forward because that's an honest debate. I mean,
when you put it out on the table, this is what we're actually doing.
We're not talking about clumps of cells and all the other nonsense that it's just disinformation. I mean, it's completely false. We're now actually debating something that I mean, I still think there's only one clear answer to this question, but it can be debated. I mean, we do deny rights to people based on all sorts of criteria.
Well, I mean, of course, and we deny responsibilities to people because they're not able to absorb them, which I don't know where I'm going with that comment, but that's I mean,
are you hopeful about resolution? Not necessarily the abortion debate. I don't want to get political either, but these kinds of things that really are scientifically based, but also with all of the complications that come with human nature and all of that is it sounds like you think we are making some progress or could make some progress because there are unrecognized areas of agreement.
I think I am optimistic that in the end, the battle has already been won. We have the resurrection.
Right.
And as much as it doesn't feel this way, we are in the end game. The forces of darkness will be defeated. And we just have to play our part. So maybe my part is I like to communicate. I have a great love for the human embryo and for embryology in general and just the magic of what happens. And I have a great respect for the truth and for facts, for evidence. And I want the debate to be on that level.
Right.
And so I have seen it move more so that way. And I think that's a complicated thing. I think women have become much more sophisticated, largely due to ultrasound. People, the whole ball of cells thing really isn't working. And when people can see, what's inside there and, what it looks like. And, and people are, they get there. They get there slowly. I'll tell you another story. I had a great... I tell the story a lot because unlike the one that makes me want to cry, this one always makes me want to laugh.
Right.
I had an attorney contact me once to question whether I would be willing to testify on the topic of fetal pain in a lawsuit against a hospital for wrongful death. And a component of the lawsuit was suing on behalf of the fetus who had been inappropriately aborted.
Right.
Or so the claim went. And so you get better damages if there was pain and suffering involved, right? So he wanted to know when, in fetal development, you would have a likelihood of having an experience of pain. And the attorney was very uncomfortable contacting me. I think uncomfortable with the case in general. But spent a long time propping up his credentials as a feminist and pro-women, and he had been an abortion litigator for decades and prosecuted dozens of cases, and he has the firm conviction that a woman had a right to an abortion no matter what at any time, under all circumstances, blah, blah, blah. And I'm like, okay.
Do you have any idea who you're talking to?
Doing this case, so something changed. What was the change? He goes, well, I got married, and we had a kid. And I saw the ultrasound at 20 weeks. Did you know they have fingers at 20 weeks? And I'm looking at this, I'm like an embryologist.
Right.
And besides that, you're a JD who has spent, by your own admission, a decade prosecuting, and you never bothered, never once, to actually look at like a high school textbook of human development. To know that, they have fingers at 20 weeks. Are you out of your mind? They have fingers at 5 weeks. But it was news to him, and he was an educated person. And a person who had ample motivation to look into the facts. But what compelled him to finally do it? An emotional reason.
Right.
An emotional reason. He had a motivation of curiosity about himself and about his own progeny.
Right.
And he wanted to know something about it for the first time. And for the first time, it was decoupled from the assumptions, from the firmly held convictions he had about a woman's right to abortion at any time. He actually needed the facts. And so that is where the truth comes out. When people recognize they need the facts.
But that there's some room for both the facts and the emotional side, the experiential side. Is that right? Maybe that's the real challenge, to find that happy middle
even
ground.
more complex than a philosopher and a scientist writing a book together.
Exactly. But it does give one hope that conversations can be held that combine the two. But our culture and everything about fundraising by groups and activists and all of that, they militate against those kinds of more complex conversations.
I think, but we can promote it. We as humans need to spend more time listening and asking questions than lecturing. Mm-hmm. I think when I encounter people who have very, very different views than I do on this, my first question is, can you help me understand why?
Yeah.
Why you feel that way about this?
Yeah.
What is motivating you, and how aware are you of the facts? And did the facts actually change anything?
Yeah.
If it wasn't evolved cells, would that? I mean, the fact changes. Really, would that make it any
That's
different?
The bottom line. Yeah. For some it would, for some it wouldn't.
Some it would, some it wouldn't. And this is actually why, as much as I really don't think it's a good argument scientifically, the fetal pain question is so compelling to people because it appeals to their sense of empathy and to their sense of emotion.
Yeah.
And so even if it's human and it was anesthetized, would you care if it was? No, it's not feeling pain, right? I mean, would that be a problem? I would still have the problem because of the human part.
Yeah.
I wouldn't feel good about. I don't feel good about capital punishment just because prisoners are anesthetized before they're put to death.
Right.
Yeah.
I don't feel good about it because I don't think we should be killing humans.
Right. Yeah. But it's not something that if we have any other option, we should be doing.
Right.
Then we have other options today.
It's interesting that the fetal pain argument tends more toward the emotional side of the conversation, and the heartbeat argument that's now prominent in some state debates, I think aims more toward the factual side.
I think they're both. I think the heartbeat argument, because it's so much earlier in development, is trying to counteract the ball cells things.
Yeah.
It's pointing to, as we talked earlier, the kind of concept of an organism, and what is an organism except something that is self-directing its own maturation. Yeah. And the fact that one of the earliest physiologic needs of the developing embryo is to have an adequate circulation, so you turn on heart development really, really early in the embryonic process. Yeah. So by 24, 25 days in human development, you have heartbeat. Mm-hmm. And it's very primitive.
What time is implantation?
Implantation is about 6-7 days.
Okay. So in that fairly long span of time in between there, is it really kind of like the mother's blood pressure driving? I mean, I'm sorry. How do the nutrients, the oxygen, is it simply able to diffuse across the?
So you establish a very primitive placental circulation, an exchange. I don't want to say circulation. An exchange between the mom and the developing embryo right away. So by a few days after, even immediately as implantation is happening, you start that process. But at that stage, the embryo is so tiny. Passive diffusion does a lot.
Yeah.
So you don't really.
Your surface area to volume ratio is nice and high.
Yeah, very, very, very favorable to. Yeah. So and you start developing an actual placental circulation driven by a very, very primitive heart in the embryo right away because growth just demands it. You start requiring a more efficient way of getting oxygen to all the bits and getting all the bad stuff out.
That's fascinating. Yeah, and that's the beauty of talking. That's the beauty of doing this and talking to people in radically different scientific subfields. Yeah. So I, of course, have the personal story about twinning that my mother's parents were both identical twins.
Both mom and dad?
Yes, her mother and her father were both identical twins, and the other members of the pair also married each other. So my mother's family is this big genetics experiment they conducted during the war.
Wow.
And to realize that is the process that happened to both of them, both pairs of them. Oh, okay.
Any other friends in your family?
Identical twins on my mother's side. I'm not sure. I know a pair on my father's side. I mean, they're second cousins that I just happen to know better than my 300 other second cousins for very random reasons. But not. I don't. Yeah, they haven't had any.
There's always been a conventional belief based largely on lack of cases that there's not a genetic component to identical twinning.
Right.
And a recent scholarly review of medical cases has presented a few families where there does appear to be a genetic component. But it's very controversial at this point. So I'm always on the lookout for familial
I do not
twinning running in families.
Neither my mother's family, my mother, her siblings, nor her genetic siblings have of their descendants. I'm not aware of any identical twins. But so I always think about that, and now I know that it was not my I shouldn't blame my high school biology textbook. It probably just didn't say. But I always just assumed, the two cells just split apart. No, it's much more exciting than that.
It is, if you watch the videos, it's very compelling process. But there's a lot of evidence that the cells of the early embryo are very, very sticky, and they're constrained within this protein code, this chemical that we talked about. People have directly tested this idea by through manipulation, making in mice binovular eggs, so two mature eggs inside the same zone of Palooza. And then you fertilize them so you get fraternal twins. And what happens is they fuse into a single twin.
A chimera.
Wow. And in fact, when we make chimeras in the laboratory, we do it exactly that way. We take early embryos, dissociate their cells, put them all back together again, and now you get a single individual coming out of those fused early embryonic cells. And this actually happens also, in the wild, as you say. Right. We do get chimeric humans, sometimes even fraternal, a male and a female twin who fuse to produce one chimeric individual who has 50% male cells and 50% female cells. So that and many other lines of evidence, and the fact that the negative evidence of never observed twinning at the 2-cell stage actually happened. All strongly suggest that even if, for some reason, the first 2 cells decided they wanted to go off independently of each other and make their own embryonic trajectories, they would re-nail back.
Because they're confined inside that.
In the zone, they would just...
The tendency is toward fusion. The tendency is not toward fission.
Right. And especially because at about the 16-cell stage, there's a change in the property of the early cells of the embryo that requires them to adhere to each other much more tightly. So this process is called compaction. And you can actually see it happen. If you watch a movie, you start out with something that looks kind of like a mulberry, and then it all just clumps back into a big clump. And the biologic function of this, to make a teleological argument, is that you establish tight junctions among all of the cells of the early embryo, and that allows for unidirectional fluid transport into the inside. So now this little clump of cells blows up into a blastocyst, sort of this fluid soccer ball, and allows for the segregation of cells into those first 2 developmental pathways: the cells that are on the outside that are going to contribute to the placenta, and the cells on the inside that are going to contribute to the placental body. So that process of compaction almost precludes the possibility of twinning until you get out of the zona. Because those cells, even if there were 2 embryos inside, once their cellular properties change and they adhere to each other, they make a single entity.
They would glue back together and be a non-chimeric chimera.
Right, they would be identical chimera. They would, we would still have the philosophical questions of what happened to the 2 souls and but I talk about chimeras in my book too.
There you go. Yeah, we'll definitely make sure to put both of those, list those in the liner notes.
And does that form part of some scientific basis for arguments in the political realm that we know of as LGBTQ and multiple genders?
Oh, gosh. Oh, man. I think we were not going to talk about that.
I'm sorry.
There's the biological side of, if you have a male-female chimera, how does that manifest in terms of their phenotype once they're...
It depends in part on the ratio of cells that are male and female. So the broader question of, is there an actual biological basis for people's different kinds of gender identities? There's a very small population of individuals who are what we call intersex disorders. So people who actually have either genetically compound sexual identities, so they might have translocation of the Y chromosome to the X chromosome, and so partially male-female genotype, or many other variations on that thing. They may be hormonally intersex, so they might have normal male genetic sex, for example, and a fairly well-known intersex disorder is called androgen insensitivity, where you have normal male sex, genetic sex, so your XY, normal number of chromosomes, everything is normal on the genetic side, but you make normal amounts of hormones, but you have a mutation in the receptor for those hormones, for the male hormones, and so your body is insensitive to the production of male hormones.
Wow.
And as a consequence, you develop as female.
Right.
Wow.
So these are women that look almost completely normal women, but they have testicles and a genetic sex of a man.
Wow.
They have testicles embedded in their
Yeah, the testicles
abdomen.
don't descend.
Yeah.
So they usually have to have them surgically removed because there's a high chance of cancer for testicular cancer at high temperature, so at body temperature. So, which is exactly why testicles normally live where they do.
Yes.
So,
Yes. Reasons for all of these things. Yes.
So,
That's so bizarre. That's a strange little detail. Yeah.
But there and then many variations on this thing. So there are individuals who actually do have ambiguous sexual identity, or conflicting, because development of normal sexual anatomy is a really, really complicated.
Yeah.
I mean, the medical students is always the most challenging set of lectures that we do with human development, because you have your genetic sex, you have your hormonal sex. So if you are genetically female but you have male hormones for a variety of reasons that can happen, you develop the male. And then you have your phenotypic sex, so what you look like on the outside, which has to do with a different set of hormones.
Yeah.
So any one of those three components can be in disagreement with the others, and then throw into the possibility that chimeras can exist in people who are mixtures of male and female cells. Mosaics can exist where, if you have a normal male embryo, and normally when you divide, every cell gets exactly the same kind of chromosomes. But if you make a mistake early on in development, and one cell of the two that are produced from the cell division gets both Y chromosomes, then you would have a 45X in the remaining cell. That's a female cell with Turner syndrome. And you'd have 47XYY,
which is
in the other, which is Edwards syndrome.
Edwards, not Klinefelter. I forget what time. There's too many of them.
There are too many of them, and I forget them.
And they're completely arbitrary, just like mineral names
some
are.
guy who discovered this the first time. But and then half of the cells would be normally genetic male cells, half of them would be male cells with an extra Y chromosome, and a quarter would be abnormal female cells with no second X. So that person would be a mix of genetic cell types, but if they are all from themselves, they didn't arise from fusion with somebody else.
Right. Wow.
So there are lots of ways that you can get intersex disorders, but the vast majority of people who have gender disorder, different, range of gender identifications, are people who are physically completely normal.
Then all three of those, genetic, hormonal, and exterior, are therein. Those are in agreement. It's a psychological. That's a different question. A very, very different question. So, whether there's then a biological basis to that psychological perspective, that I think we're still out. The jury is still out on that one. Yeah, and the atmosphere that we live in is not conducive to studying that with a disinterested quest for the actual facts. I can hardly imagine.
Yeah. Well, I don't know. I think it's a curious thing whether or not the LGBTQ population would endorse that kind of a study or not. I mean, it used to be for many, many years, for example, there was a push to find a gay gene,
Right.
something that allowed homosexual individuals to say, well, look, I was just born this way. It's genetic.
Yeah.
The community has kind of moved against that now, and because they don't want to believe it's deterministic. They want to believe that gender is fluid and people can pick whoever they want, and it really has nothing to do with biology. It's people are free to have whatever orientation you like, and
how dare you tell me that biology is destiny?
Right, kind of like that, yes. Yeah. And I think that's much more the dominant view right now, that there's much less of a push to try to find a biological basis for people's orientations and identities than just simply accepting that they exist. And I have a right to have it if that's what I want.
Right. Well, I have a right to have it without consequence X, Y, or Z, and with support P, Q, and R.
We've got kind of far afield on this one.
Yeah. Well, that's
fascinating.
I mean, we have been here for an hour and a half. You've been very indulgent of letting us continue to ask questions and extend the. It will always be a red letter day for the reasons I mentioned in terms of, I learned how identical twinning actually works today.
Yeah,
That really, I'm still kind of reeling from that.
That is my argument. I'm not sure if you would agree, but I think it's a very sustainable argument.
Yeah.
And I think it actually solves a lot of the philosophical problems. Because at that point, at that late of a stage, say day 5, when I think 4 or 5 when most twinning happens, or all. You've clearly established yourself as an organism, as a developing human that's moving towards a greater stage of maturation. Yeah. And then what happens when the embryo splits at that point is regeneration. That we are very good at repairing ourselves from injury, and so you get a catastrophic injury, you lose half of your body, and you replace it. Yeah. And the fact that each of the halves does this is where people's brain starts bending. And, so who's the original twin? And, but if you simplify the question, if you split an embryo in half and you throw away one half, and watch what happens to the surviving half. The surviving half acts like any organism acts when it's wounded. It repairs itself. So it tries to reestablish the original number of cells that were there in each of the various parts of the embryo at that stage of development. It heals itself, it reseals back into a ball. And if you watch the development of this half embryo compared to unsplit sibling controls in an animal model, it advances exactly in synchrony with the unsplit twins. So there's not even a pause. It just keeps running. It keeps doing exactly what it was doing before you split it. Wow. And it's just half the size. Yeah. And that argument to me, or that observation, would very strongly support the conclusion that you have a whole embryo, then you have a damaged embryo, and then you have a regenerated, a healed embryo. Yeah. And it's the same embryo, because it started along this process at spermic fusion. Yeah. It set up all the things that it's doing now, and it continues doing them without any perceptible pause. So my conclusion from that would be splitting an embryo at that stage constitutes an injury, and the embryo regenerates, recovers from that injury, but it's the same individual throughout that entire process. Okay, so what about the other half? Right. Yeah. Now, the status of the other half, the status of the first half we just considered, is in no way dependent upon the as of yet undetermined status of the second half that we threw away. I mean, logically, they're completely separate. Yeah. So now when we turn to the consideration of the second half, there's also no evidence that the original embryo died. All we're left with is the challenge of knowing which of those two twins is the original and which is the newly generated sibling. And the fact that we can generate a sibling at that stage seems unfamiliar to us, but it's well within what happens in biology in many species. Really? Yeah, asexual reproduction. You can chop an earthworm in half and you get 2 earthworms that are genetically identical to each other, because both of them heals itself and continues on. And no one would argue that there are two, that single earthworm is not an individual at the beginning, or that there's somehow 2 earthworms packed inside of one, and just because it could get cut by my shovel when I'm gardening. Right. Yeah. So the same thing is true of us, and I think that the most reasonable interpretation of twinning is that one of the twins is the original. One of them is a newly generated individual that comes into existence at that moment.
Yeah. I will definitely be, again, like I said, with my intrinsic interest in the subjects for understanding my own history, definitely be looking into that book. So,
great.
I think we should look back.
Yeah, I think we should.
Yeah, thank you so much.
Cut that there. We appreciate it. That's probably 3 episodes worth of material there. So that's a wealth of.
Have fun.
We don't usually trick guests.
I don't. I think it's the first time we've done that. But,
it's the kind of thing that just. Once you get started, it's then there's this, and then there's this.
Yeah.
But this is why biology is just so fascinating, because it is, to some extent, self-exploration.
Yeah.
I mean, it's one of the fields of science where you're really asking questions about your own nature, about who you are.
Yeah.
And I think that compels people. Whenever there's a book, I must say, anyone who has twinning in their background is always more interested in twinning.
They're always more interested in twinning, yes. You can identify that. Yeah, exactly. Yeah, I'll read it and then I'll send it to Katie and Christy because they will probably,
Yeah, it should. I wrote the book. I'm hoping for kind of the educated non-scientist. It's you gotta be a little fearless and be willing to. Okay, I didn't understand that word, but I'm just gonna flip over. Yeah. There is a glossary in the back.
That's what those are for.
And this non-scientist will read the one where the philosophers in dialogue.
Oh, that most assuredly that one too.
Yeah, definitely. If that book is worth purchasing just for that first chapter, that really goes through how moralism and what does it mean, and tries to explain why this isn't just a made-up idea. But if we don't have this, we can't do science. We really cannot. Intrinsically, scientists understand it. They don't recognize it, and they call it hocus pocus.
Right.
But it actually is necessary for us to do what we do.
Yeah. Then you can either have it out consciously in front of you and be able to refer to it when you're in difficult situations, or you can just have it unconsciously in the background and stumble through and
then criticize anybody who actually has a better understanding than you do.
Right? Yeah, always that. That's human nature in a nutshell. Well, okay.
It is.
Yeah. All right. Well, thank you so much.
Very, very welcome.
If you enjoyed this episode, or it made you think, come on over to That's So Second Millennium's Facebook page and leave a comment or ask a question. We'd love to hear from you. Our intro music is Mars, the Bringer of War, and our outro music is Venus, the Bringer of Peace, from the Planet Suite by Gustav Holst, performed by the U.S. Air Force Heritage of America Band. The recording is in the public domain and made available by museopen.org. For my co-host Bill Schmidt, I'm Paul Giesting. Thanks for listening to another episode of That's So Second Millennium.